The title of today's webinar is Foodborne Pathogen Surveillance with Oxford Nanopore Sequencing. Our speakers today are Andrzej Benkovsky, Senior Technical Manager at Eurofins, Romain Chauvet, Molecular Microbiology Lab Manager at Eurofins Laboratoire Microbiologie West, Jessica Chen, Team Lead, Bioinformatics Research and Development at the Centers for Disease Control and Prevention, and Peyton Smith, PulseNet NGS Activity Lead at the Centers for Disease Control and Prevention. With that, I'll turn it over to Andrzej.
Thanks everyone for tuning in today. Welcome to our webinar on harmonizing global pathogen tracking with ONT sequencing. My name is Andrzej Benkovsky. I'm joined by my colleague Romain Chauvet from Nantes Laboratory in France. And today we're going to share some insights from our proof of concept and discuss how Eurofins is driving global harmonization and WGS for food safety. So what we're going to cover today, first, just kind of setting the scene with the role of whole genome sequencing in food microbiology, then go over what we did for our proof of concept for our global collaboration, followed by Romain discussing more details related to the methodology and the bioinformatics involved. And then we'll do a little bit on results of the proof of concept, the verification, and discuss the global impacts and future directions. So, as we probably already all know, WGS really has transformed food microbiology by providing unparalleled resolution for pathogen ID and source tracking. And the advances in sequencing technologies have significantly reduced those costs and turnaround times. And really this makes WGS more accessible for routine use in third-party testing labs such as Eurofins. You can see the test cost decreasing on the charts to the right, and then already a substantial $2.1 billion market share globally. The market's only expected to grow with 11% compound annual growth rate through 2030. So traditional methods like MALDI-TOF and PCR provide genus or species level identification, but really they lack the discriminatory power needed for that strain level resolution. And when we're talking about source tracking, really we want to get down to the strain or serovar level. WGS eliminates the ambiguity of species and genus identification, and really that's critical for differentiating the closely related strains. And this level of precision is now really considered the global gold standard for microbial taxonomy. So WGS for source tracking, really when we talk about source tracking, we're talking about genetic variations such as single nucleotide variants, insertions, deletions, structural rearrangements, and by comparing the number of S and Vs between different pathogen isolates, we can quantify genetic proximity and determine whether strains share a common origin. And really that's the key when we talk about identifying sources of contamination and performing those types of investigations. And really this approach provides that robust framework for linking contamination events, specific sources within those complex supply chains. So while whole genome sequencing offers powerful insights, it's not necessarily a plug-and-play solution. The methodologies can vary across regions due to the different regulatory requirements. The rapid technological evolution, as we've seen with Oxford Nanopore technology, and of course the diverse objectives, different groups using whole genome sequencing for pathogen tracking. When it comes down to it, standardization is really going to be essential to ensure that the data generated in different labs and different groups are comparable and actionable. And really, this is the foundation for Eurofins' global harmonization strategy. So Eurofins as a whole is over 900 laboratories, but within the food and feed division, we're a global network of over 200 labs, and we partner with multinational food manufacturers to deliver standardized monitoring tools. Our recent proof of concept study that will go into more detail demonstrated that a laboratory in the United States in Madison, Wisconsin can perform wet lab sequencing, share FASTA files with the team in Nantes for bioinformatic analysis, and this model eliminates the requirement to ship pathogens internationally. This, of course, reduces turnaround times and enables our global clients to maintain consistent pathogen surveillance. So our objective, implement a globally harmonized ONT-based whole genome sequencing method for key pathogens such as Listeria, Cronobacter, and Salmonella. By 2026, we expect the standardized approach to be deployed across Eurofins labs in the United States, France, Brazil, and China. And this unified validation framework really is going to ensure that reproducibility and comparability we're all seeking. And this is going to support our multinational and global food safety programs for our various clientele. Okay, next I'm going to turn it over to Romain, who's going to discuss a little bit more about the methodology and the bioinformatics.
Good morning, good evening to everyone. Thank you, Andre, for the first part of the presentation. I will just present 3-4 slides for technical parts, and after that I will give back the mic to Andre. So to go a little bit more in details about methodology, how we perform whole genome sequencing in this harmonized approach. It's very basic. It's more or less what is recommended by Nanopore. So in 6 steps for the wet lab parts, first, of course, start with morphotype verification, because if the customer request is to track Salmonella, you just have to make sure that it's Salmonella on the go plate. It's not so easy sometimes to differentiate between some Cronobacter and some Salmonella, for example. So we always start morphotype and we move next for subculturing to be sure that all cells on the GAR plate share exactly the same genome. It's very important because it's one of the biggest hypotheses for bioinformatic analysis after that. When the culture is ready, we move to DNA extraction, purification, and fragmentation of DNA. So again, it's very common. It's beading for cell lysis. It's purification on silica column, and we use G-tube for sharing. For the library preparation, we use here the kit from Oxford Nanopore NBD, the last version, 114. It's quite labor intense, but at the moment, it's the only option to collect data set that can be used for SMB calling. With this kit, of course, there is multiple steps, for quantification, for normalization, and so on. So yeah, it's quite labor intense, but at the moment, it's the best way for multiplexing and to get very high quality data from Nanopore platform. We load the library into a flow cell. Here it's R10.4.1, and we let the sequencing run for more or less 1 night to get sufficient data for each sample. When everything is done, we move to bioinformatic analysis. And here, typically, it's when we are tracking pathogen, we move to SMG calling approach. So again, we recommend Nanopore. I mean, you can recognize each step of the recommended workflow. So since the thing with GUPI barcoding, and of course we do some check, and one of them is to check the quantity of raw data for each sample. We move to filtration. So filtration is, as usual, based on quality and size of reads. And again, there is some quality check, and everything here is done with nanofield. So again, tool recommended. When everything is clean, we start to assemble the data set. So with the long reads, it's very easy to go with the de novo approach, which is very convenient for everything at this time and after for SNV calling and database management. This is very big advantage of Oxford Nanopore here. It's to make de novo assembly very easy. So there is some quality check, of course, like continuity of the genome or N15 and identity, of course, of each context if there is many contexts. For customer perspective, we had an extra step here. It's database management. What we do in Eurofins is to work as customer-specific database comparison and not comparison. It means that database for each customer, and we will feed the database that there is a capitalization on the data and on the tracking approach. When the database is clean and clear, we can start SNV calling. So all strain will be compared together within a database, a customer-specific database. And again, we use a Nanopore-related tool, NanoCaller, which is quite complex with a lot of parameters, but just had an extra city region with high mutation. It's most of the time related to genetic mobile element, which is not relevant at all for tracking of pathogen in the food industry. Next, we have a classification approach. So because for some customers, the size of the database is start to be very big. We have reached more than 1,000 isolate for some of our customers. So at this size, it's just impossible to interpret the result based on the SNV table distance. It doesn't make sense. It's hours for scrolling and just finding some trends and so on. So what we do is just to define some threshold and classify strain according to this threshold. And after that, of course, we report with document with everything inside, methodology, and of course, some guidelines to interpret and so on. So this is how we perform tracking of pathogen with whole genome sequencing approaches and of course, SNV bioinformatics. And to be sure that for our customers, whatever the place where they decide to work with Eurofins, because some of our customers are working globally. They have some plants in USA, in Brazil, in Europe. We want to make sure that the data sets are always drilled to be consistent, whatever the lab. So we try to design an experiment to validate this approach. So first we decide to harmonize together, and this harmonization was from the beginning to the end of the workflow. So we try to validate an end-to-end approach. It means that we, of course, start from cultivating the microorganisms to DNA extraction, leaf prep, sequencing, and bioinformatics. So we keep aligned everything on this workflow, including, the version of the software and so on. This experimental approach for proof of concept was done over 6 months to be sure that we catch variability, like, different batch of kits from Oxford Nanopore or from the suppliers to be sure that it's relevant in times. And we just use 2 strains. It's a strain very common in the food microbiology labs, food testing microbiology labs. It's just collection strain, just to make it easy for everyone to have strain in the labs. And just to keep it short, but we always sequence the first strain when we start sequencing just has a positive control. So it's a systematic positive control as well. So it was the case during the study, but now in production as well. And what we have done to estimate if the proof of concept was good enough for production or not, we defined 3 criteria, performance criteria. The first one was repeatability. So it means that it was just in one lab, or in each lab, sorry. That is the same strain, the same batch, the same sequencing run, and the same technician, just 3 independent culture of Salmonella arizona, just to estimate how much assembly we can detect between 3 independent cultures. And after that, we introduce potentially more variability, so inter-lab reproducibility. So it was just replicate, but not in the same batch, not in the same sequencing run. And after that, of course, for this proof of concept to be able to prove that we can perform the same analysis and to get the same result at the end, we perform inter-lab reproducibility, sorry. So the same strain here, but just 3 different labs. So everything is different, just the strain are the same. Very basic. This is what we get. Sorry. So the first column is not very useful here. It's just internal code for us to find samples. The second column is description. So the name of the strain, the name of the lab, the 2 letters, US for USA, BR for Brazil, and FR for France, the date of analysis, and the replicate number. In the middle of the table, you can see some 1, 2, 3, 5 columns, just about metrics and some quality check. Just some reported. There is more quality check, but just for the purpose of this presentation, we reduced 5 here. So it's purity, it's reading death, and 50, the number of contigs, and the percentage of homology based on the ribosomal region. And on the right panel, of course, you can see the assembly table, so pairwise distance table. So almost all of comparison were done between DSM strain. Just the last one was added to give more variation in the data set. So you can see that it's not a perfect zero. There is sometimes zero mutation detected between data sets, but it's more or less between 6 and 0. If we go a little bit more into details, for the quality check, the first quality check, it was quite selective for one sample, so it was needed to rerun one sample because the quantity of data was not enough to have sufficient coverage. It's not uncommon because sometimes it's a little bit tricky to do a great normalization of the library, and sometimes one sample is a bit imbalanced. So just with the rerun, it was arranged and it was possible to add this sample to the data set of comparison. All culture, of course, were confirmed as Salmonella. It's great. We are a microbiology lab, so everything works well. And even if what's not shown in the table, we performed several predictions with SEC02 and everything matched accordingly. Considering the data set after processing, just on the assembly level, the minimum coverage was more than 54, but in average it was more around 200, which is quite big for such application. And as I said in the previous slides, Nanopore is great for de novo assembly, and it's very easy to have almost complete assembly at the end. So it was more or less a perfect assembly for almost all samples. So in the way, 2 contigs, one for chromosomic and the other one for the plasmid, and that's all. It's great because it makes possible to perform a true whole genome sequencing comparison because when we perform reference-based approach for compare some samples, always a part of the genomic information is missing. Depends on the representativeness of the reference genome used. So here it's not the case at all. There is no reference genome, just a direct comparison of genome in the database. And considering SNP analysis, so based on the table shown before, the within lab repeatability is less than 1 SNV on average. The interlab reproducibility, again, is less than 1 SNV. And interlabs reproducibility is, in average, less than 1. And we have seen a max difference for 1 or 2 pairwise distance. It was 6, the maximum. And at the moment, our recommendation for defining clonality, it's about 20. So we can consider that it's acceptable for what we propose for our customer. And this, of course, it's not zero. So there is some noise, some technical noise, maybe some a little bit technical, not technical, biological noise as well, because we can introduce some mutation when we perform some self-filtering. But for the food industry to take decision, this noise is very acceptable because it's almost nothing comparing to the diversity that we can have in the food processing plant. So if you want more information, just send us an email and we have a very short document that presents this result with a little bit more details. Andre, it's now for you.
Thanks, Romain. So I'm going to take you through a quick case study, how we used this new harmonized approach to help one of our clients. So we have a global food manufacturer operating more than 50 plants across 3 continents. With the increasing supply chain complexity, the risk of their Salmonella contamination became a major concern. And the issue arose when Salmonella was detected in finished products at plants in both the United States and in Italy. The cultural confirmation was straightforward, but the initial investigation faced challenges. There were inconsistent local testing methods, slow turnaround times, and the absence of standardized genomics comparison framework. So our solution was to deploy the Eurofins harmonized WGS approach using Oxford Nanopore sequencing. And by implementing the standardized ONT protocols across the multiple global labs in the United States and in France, we significantly reduced the turnaround time. And then, of course, with the central bioinformatics pipeline, this provided the SNP level resolution for then an accurate strain comparison against the secure client-specific database. And that really ensures the traceability and data integrity associated with the testing. So the implementation was rapid, efficient. Salmonella isolates from both plants were sequenced locally within 48 hours, and this eliminated the delays associated with international sample shipping. The analysis ended up revealing that both isolates belonged to the same clone. This pinpointed a common supplier as the contamination source. Corrective actions included a supplier audit and targeted hygiene improvements, and the results were significant. The root cause was identified in just 5 days, compared to sometimes weeks typically required for more conventional identification methods. This response helped avoid a global product recall, saved the company an estimated $15 million, and helped establish this global pathogen surveillance program for their network of facilities. So while this harmonized whole genome sequencing offers clear benefits and industry-wide adoption, still faces some hurdles. These include the need for the upfront investment in the infrastructure in order to perform the testing, training the personnel and the expertise required to perform the analysis, and then aligning with the evolving regulatory framework across the globe. And our key takeaways are that Harmonized WGS enables rapid, reliable, and globally comparable data, while local sequencing combined with centralized analysis dramatically reduces turnaround times. And then looking ahead, we're going to continue expanding the NGS laboratory network, covering additional regions, additional pathogens. We're also pursuing validation and according to ISO 23148, as well as ISO 17025 method accreditation. And this will help strengthen our compliance and quality assurance. And really, ultimately, our goal is to make a proactive genomic surveillance a standard practice for global food safety and brand protection. So with that, I'd like to thank you. If you have any additional questions, we'll have a Q&A at the end of the event. And I appreciate your time this morning. I'll turn it over to the next speaker.
Hello, greetings. My name is Peyton Smith. I'm happy to share with you today about the PulseNet ONT evaluation and getting us started. I'm seeing buffering on the slides. Let me find it next here. Can you refresh it? Oh, there it goes. All right, there we go. So the format for our presentation here, I'll get us started with an introduction to PulseNet USA and PulseNet International and our interest in the ONT platform. And then my colleague, Jess, will take us through the technical breakthroughs with ONT and the PulseNet USA validation. And then I'll come back and talk about PulseNet International and the impact of ONT on that particular network. So starting off with an introduction to what is PulseNet USA and PulseNet International. So in the United States, 1 in 6 Americans will get a foodborne illness every year. And a subset of those people may then go to a doctor or a hospital where a doctor may order a stool sample to be taken. That stool sample is then worked up at a clinical laboratory. And if that sample has one of these common foodborne pathogens, like on the slide here, identified, then that clinical lab forwards a sample of that bacteria to a public health laboratory, where at that public health laboratory, whole genome sequencing is performed on that bacteria, and that sequencing and molecular subtyping data is then shared with PulseNet USA. So, what is PulseNet? PulseNet is the national molecular subtyping network for foodborne disease surveillance for disease-causing bacteria. PulseNet USA is made up of 82 state and local public health laboratories, as well as food regulatory agency laboratories, and is coordinated by CDC and by the Association of Public Health Laboratories, APHL. And basically, how it works is that these 82 laboratories in PulseNet USA all perform whole genome sequencing using standardized sequencing procedures, and then share that molecular subtyping data to CDC HQ here in Atlanta. And at CDC, those molecular fingerprints that are shared are then analyzed. There we go. Are analyzed to try to find clusters of illness with the same molecular fingerprint. Sample or clusters that are found with the same molecular fingerprint can then be communicated with epidemiologists, who then may go on to initiate an outbreak investigation to find a common source. There's also the PulseNet International Network, which is grouped into 8 different regions around the world and has over 80 participating countries. The real focus of PulseNet International is to provide support on technical assistance, to provide technical training, and to facilitate regular communication and data exchange between laboratories and across countries to find sources of outbreaks, look at AR surveillance, and work together. And why PulseNet works is because it's built on these key elements of the PulseNet framework, from PulseNet laboratory capacity building that involves using standardized training procedures and materials across PulseNet USA and PulseNet International, a very robust quality assurance program that includes standardized procedures both for the wet laboratory and the dry laboratory, a certification program for participants of PulseNet, and an annual proficiency testing program and certification for PulseNet as well. Because we're doing whole genome sequencing on isolates right now, we also incorporate surveillance of antimicrobial resistance from whole genome sequencing data, which is important to track and trend emerging AMR that could be causing a problem. And then last, a key part of PulseNet is to strengthen communication between the laboratory and epidemiologists to ultimately improve the process of going from data to action and hopefully stop an outbreak. But also important to PulseNet is cost, because this is a public health driven network. Cost, and we're all operating on a narrow budget, cost is an important factor. And this figure we see here are the costs per isolate or cost per sample when performing whole genome sequencing using the PulseNet procedures going from DNA extraction all the way through sequencing. And the bottom line I wanted to point out here is that the good news is, looking ahead towards the future with the platforms that are already being used in PulseNet now or being evaluated for use in PulseNet in the future, is that the cost per sample is going down. And specifically, we're talking about ONT today, which when sequenced efficiently can be under $50 US dollars per sample. And the reason PulseNet is really interested in ONT are because of these things here, starting with accessibility. There is a lower cost of entry for using ONT sequencing, using a Minon device. It has a smaller footprint, and the portability make this technology very accessible genomics around the world. The cost effectiveness, like we saw on the last slide, the cost per sample is quite good. The ability to analyze sequence data in real time has a lot of potential. And then last is the ability to use those long reads in ONT sequencing to produce very high quality de novo assemblies of bacterial genomes and get that very valuable genomic context. So with that, I'll hand it to my colleague Jess, who will take us through technical breakthroughs.
Awesome. Thank you, Peyton, and thanks everyone for joining today. I'm going to walk you through some of the technical breakthroughs that have occurred from Oxford Nanopore, both on the sequencing chemistry side and the bioinformatics side, that made it possible to get to where we are today. So we've checked in with ONT sequencing and used it periodically over the years, but we were really excited in 2023 with the launch of the V14 kits that had with the R10.4 flow cells that were delivering Q20 plus chemistry. This was a huge advance in accuracy, and accuracy is incredibly important to us because if this is a technology that we're going to onboard in PulseNet, it has to be accurate enough for us to run our outbreak detection on. And so briefly, I'll talk about kind of the basis of how we compare genomes in PulseNet. Our standard methodology is using core genome multi-locus sequence typing. We use CGM-LST for most organisms. There are a handful of organisms where we use a WGM-LST scheme because that's what's needed to provide the resolution as well. And just to briefly go over CGM-LST, core genome MLST is the core genes are those genes that are common and shared amongst 95% of isolates for a single genus or species. And so usually, depending on the scheme, we're interrogating about 50 to 80% of the genome in this type of approach. The accessory genome are those genes that are found in only a few strains. We do look at the accessory genome in some of our genotyping approaches, like Peyton alluded to, looking at AMR. We also in PulseNet look at virulence genes and do a lot of other tests, but CGM-LST is kind of our our focus in this validation, since it's what we need to compare genomes and detect outbreaks at the surveillance level. So core genome MLST works a lot like, 7-gene MLST. If you've used that, it's just on a much larger scale. You've got a scheme of conserved loci across the genus or species, and you identify the individual alleles at that level. So when first taking a look at the Q20 chemistry, we noticed early on that most of the differences that we saw in the genomes were at these bacterial restriction modification sites. So a little bit on bacterial restriction modification systems, so everybody understands it's a bacterial defense system to protect against foreign DNA. So there's a nice figure here that shows a phage injecting its foreign DNA into the bacteria. And DNA restriction endonucleases cleave that foreign DNA as that defense mechanism. But the host wants to protect its own DNA, so it uses methyltransferases to methylate host DNA at these specific sites. So it's at these specific sites where there is methylated DNA present where we were seeing discrepancies between ONT and short read technology. But in September 2023, we were really excited when ONT released a bacterial-aware base caller. There had been base calling efforts for like human DNA that was aware of those methylation modifications, but those are different for bacteria. So we were really excited to see a bacteria-focused tool. This was initially called Ririo, but all these models are now worked into Dorado. So that's what we use going forward to address any of these, to address the base calling. So to give you an idea of the impact, sometimes when we sequenced, we would see very few differences relative to Illumina, but with other strains, because of methylation, we were seeing upwards of 25 to 39 alleles. This is one particular sample of Shiga toxigenic E. coli. However, the Dorado bacterial methylation brought us down to 1 to 2 differences. So this was such a huge leap for us in being able to improve our accuracy and really hone in on our outbreak of interest. So here's kind of a zoom out, not just looking at one isolate, but looking at several isolates across the same outbreak. You have on the left here the outbreak cluster and these closely related outgroups beneath it. So this is just a heat map of the different pairwise allele differences between isolates in this outbreak. And you can see you have a really tight outbreak cluster of 0 to 2 alleles, whereas your closely related outgroups are clearly distinguishable. So this was really exciting for us to see, and we decided to kind of then ramp up our evaluation efforts. Another thing that we really helped us out along the way were improvements in consensus polishing. So there was a big leap for us from Medaka 1 to Medaka 2. So you can see this is our kind of standard benchmark 30-strain panel that we use to evaluate any new tool or library prep initially. So we're looking at Salmonella, some Campi, Shigella, and E. coli. And you can see that in Campi in particular, we were seeing some larger differences in the kind of 10s to 20s amount of allele differences, where in Salmonella and E. coli, we're seeing much smaller differences. But with the Medaka 2 consensus polishing, coupled with the improvements in base calling, we started seeing that number shrink drastically. So across the board, we're only seeing 0 to 2 allele differences in this particular data set. We have, on our side, developed an ONT assembly workflow that we're optimized we have optimized for a cloud platform, PulseNet 2.0. This is a fairly lightweight workflow with kind of a streamlined set of tools that you can upload a batch of sequences. It runs QC, some downsampling and filtering. It then assembles with Fly, and then does some downstream post-processing after that assembly, kind of orienting the chromosome to a specific start, does the Medaka polishing, and does some downstream QC using Busco. This workflow is online at our GitHub. We call this workflow Stilo, and it's currently submitted to the Journal of Open Source Software under pre-review. Just this summer, there were some new base calling models released around London calling, and we were really excited to evaluate those. This is that same 30-strain data set that I talked about previously. And so we've got in red models using HACK, and blue the models using SOUP, and we've got both 5.0 and version 5.2 software models for Salmonella, Escherichia, and Campylobacter. So you can see this scale is from 0 to 5, 0 to 6. And you can see the ranges on these are quite narrow. We're seeing majority of these 0 to 2 allele differences. So this is really exciting. This is also using the new, there's a bacteria-specific flag in the Medaka polisher, and that really improved the HACK models in particular for us. We got a huge glow up on the HACK performance with this, so we were also excited to see that as well. But right now we're sticking to the SOUP 5.0 base calling as our standard. We will accept from our partners 5.2 base calling, but anything in that 5.0 or later base calling is great for us, works well for us. So getting into our evaluation, this is some of our preliminary data for our large-scale validation. We're rounding this out with a couple more organisms that are of interest to PulseNet. For example, Cronobacter, some Yersinia, but right now we've evaluated some Campylobacter, Listeria. We've done the most work with Salmonella, focusing on the top 30 serotypes. We've looked at some Aztec, and in here is some Shigella as well as Vibrio. And you can see the majority of these are right under 5 allele differences relative to Illumina, so that's really great to see. We have a few outliers, but whenever we see these outliers, we tend to dig deep into these and investigate, are these methylation sites? Are these things that we can flag to the folks at ONT as something that could potentially be included in new models? So this is always an improving process for us, and I can really only see it getting better from here. We've also released to our we've also developed at CDC an SOP for our rapid barcoding kit, which is the kit that we use typically. We've shared these draft documents with our pilot states. We have a 10-state pilot where we are having them test out this protocol and use it on the certification strains that we use for PulseNet to start evaluating kind of the site-to-site variability, much in the way that Eurofins discussed their studies to look at site-to-site variability. And so to talk a little bit about our PulseNet ONT working group, as I mentioned, we've shared that initial draft proposal with the group and are soliciting feedback from them. It's a really great spot for knowledge exchange where people can share their experiences with, say, different extraction methods or what library prep methods they're using, what automation they're using, and even into the bioinformatics as well, and sharing just results and success stories. So that's been really great to hear from other users on that point. It's been awesome for data sharing. So all of those sites are sharing data with us, and we're running those through our workflows to kind of get a standardized look at how the data is looking from all the different sites, and discussing bioinformatic pipeline development as well. And we have our, as I mentioned, our multi-site evaluation testing, which is looking at that site-to-site variability using a standard set of strains. So here's a look at that comparability. We have basically one strain of Campi, Listeria, Salmonella, Aztec, and Vibrio. And each of the dots are a different site, and the x-axis is relative to CDC-generated Illumina sequence. So we're seeing kind of very tight reproducibility on Listeria and Campi. We have a little bit more variability with Salmonella, which is interesting because that's actually what we observe in our certification data for PulseNet for short read technology as well. So we don't know if it's something about this strain, but you know that in talking to some folks in the lab, this was not entirely unexpected to see more variability for Salmonella. We have really tight ranges on Aztec, and Vibrio seems to be performing really well with a few small outliers, but all in all, the kind of largest outlier we saw was 8 alleles. But again, most of the data concentrating, you know, below 3 alleles. So this is looking really nice, and we're continuing to have discussions with the sites to try to understand like why we see some variability from certain locations, but you know, like I mentioned, we did expect to see that with the Salmonella. And from here, I'll pass it over to Peyton, who will wrap things up talking about some of our efforts with PulseNet International. Thanks, Peyton.
Thank you, Jess. In our last few minutes here, I'll talk about PulseNet International and the impact of ONT in this particular network. So jumping right into it, we have done 2 trainings on the PulseNet ONT procedure. One of the trainings was for the PulseNet Africa region, where representatives from each of these countries listed here came together for a week for a workshop in the Gambia at the Medical Research Council Institute. And during this training, it was a hands-on workshop where participants got hands-on to prepare libraries, perform sequencing on the ONT platform, and then analyze their own sequencing data using bioinformatics tools, all together using standardized procedures. And I'll talk more about the data that came from this training shortly. We also did another training for the PulseNet Asia Pacific region, which was quite a large training from these countries listed here. Multiple sites for some countries. That was hosted at the Thames facility in Thailand. And similar to the PulseNet Africa training, this was also a hands-on workshop where participants got hands-on doing library preparation, sequencing, and analyzing their own sequence data using bioinformatics tools, using standardized procedures. After the PulseNet Africa training, I was really pleased to see the engagement coming in from the data that was generated during the training, looking at genomic diversity and antimicrobial resistance of Vibrio cholera isolates from countries all around the continent. And I encourage you to take a look at this preprint from Dr. Eben. And these training cohorts are kind of where they are now, are focused on these feasibility studies, where the idea behind those is that participants submitted a proposal for completing sequencing with the platform, and some of those proposals were selected, and those sites are expected to sequence the PulseNet International certification strains to become PulseNet certified at their laboratory. They're also expected to sequence 100 enteric bacterial isolates using ONT according to their proposal, which is focused on AMR surveillance and using a One Health strategy. And ultimately, what we're trying to show is that genomics can be implemented for enteric bacterial surveillance globally, and that it can be used especially for antimicrobial resistance, for outbreak monitoring, and ultimately to show that data exchange within the PulseNet International Network can happen, and that it can facilitate data to action and prevent poor health outcomes. So to wrap up here, looking ahead for where PulseNet is going with ONT, we want to finalize our testing with the validation. Jess mentioned some other species, some of the rare species seen in PulseNet. We'll look at additional genotyping metrics. We want to implement the software for ONT and integrate the platform into the PulseNet 2.0 software for PulseNet USA. We want to continue our work with the PulseNet USA working group to refine and optimize our SOPs and quality control parameters. We're going to stand up a certification process for laboratories to become PulseNet certified for ONT sequencing for those that are part of PulseNet. We're going to complete our formal validation through CDC. We're going to implement a PulseNet ONT proficiency testing or PT program. And last, we're going to continue PulseNet USA and international trainings on the ONT platform to implement this as an additional technology for continued surveillance of enteric bacterial disease. That I'll quickly hand it back to Jess.
Thanks, Peyton. I just want to acknowledge all of the folks who have contributed to this work over the past couple of years. We have a huge group in EDLB that has worked on it. That's our branch at CDC that's worked on this, these developments. And a lot of our international work has required a lot of collaboration with some fantastic partners at the Doherty Institute, the London School of Hygiene and Tropical Medicine, Pathogen Genomics, of course, our partners at Nanopore, who have been really instrumental in listening to our experiences with the platform and giving us a lot of advice along the way. We've got our folks at APHL who have helped us stand up our working group, who have been amazing. And yeah, I just want to say thanks for tuning in.